Good afternoon. Welcome back. This is lecture number 4, The Origin of Life. I'm Dr. Beal, and this is Gen Bio 115. You should have your very first workshop this week. When you go to workshop, you need to have with you BIOS forms for lectures 1, 2, and 3. When you go to workshop next week, the second week of workshop, you'll bring your BIOS form for this lecture, lecture 4, and lecture 5, which I will release on Wednesday of this week. Now, this is a short lecture. It is to make up for the fact that lecture 3 is so long, but it's also some highlights of things that your book really doesn't focus on, but we need you to focus on in order to go ahead with Gen Bio and where we're going for 116. So we're really going to look at 2 big topics here. We're going to look at abiogenesis. This word is really nice and easy to break down. A means not, bio means life, and genesis means the creation of. So this is the creation of life from not life, or the story of how life started on Earth. And then we're going to look at a history of what happened from the start of life a little bit far, not to modern day, but just some highlights. The outcomes for lecture 4, The Origin of Life, are I want you to CC the requirements for the origin of life. Give me the 2 hypotheses that I show you for abiogenesis, and then compare and contrast them. Sequence abiogenesis, what happens, what do you have to have, give me about a timeline, and then sequence the major evolutionary life events. Now, I'm going to have 2 big words in this that sound an awful lot alike. Abiogenesis. Abiogenesis is the thing that's going to help you here. This is not life to the creation of life. And abiotic are factors that are not alive. So they're close, but I want you to, as we go through, make sure you're comfortable telling them apart. And if you're asked to define one on the exam, make sure you don't mix them up. And then as flowing forward, you may not be able to do this today, but something I might ask you on the final exam is, can you give me 2 or 3 pieces of evidence that support the theory of endosymbiosis? We'll talk about that. And that'll help you as we keep coming back to this idea of endosymbiosis that we're going to introduce today. This is real fast. You can take a picture. You can pause me here. This is the description of what goes into each one of these. I'm going to do these for maybe 1 or 2 more lectures, and then I'm going to ask you to try and figure out what these mean. I left you off with a practice question, and please keep coming to my reviews. I think they've been going well. We've been able to talk about why and build the compare and contrast charts. We'll do some sequences this week about how to understand these molecules and their functional groups. This is a really basic question asking you basically, where does the peptide bond go? This is an amino acid, an amino, and a carboxyl acid. They're bound together by this central carbon. We call it an alpha carbon. And there's probably something else here. There's a remainder of the molecule. This is asking you, where does the peptide go? We're going to add a new amino acid over here. I'm going to form a peptide bond, and if I form it from my carboxyl group, I'm going to attach the amino group of the amino acid next. Bonus question 1. I'm not going to answer this today. What level of protein folding is this question asking about? Bonus question 2. What reaction is occurring when I form peptide bonds? You should be able to answer those right away. If not, go back to your notes and answer them before you come back to my next review. So we'll get started. The origin of life, we're going to start out with abiogenesis, or the creation of life from not life. This is an artist's rendition of what Earth looked like prior to life existing. It's really unpleasant. Lots of energetic volcanoes going off. It's probably, the temperature varies wildly. There's not a lot of water in the atmosphere, maybe a little bit. A lot of this pictured here is not water, but it's melting soil and melting metal. There's comets striking. There's no oxygen available in the air. This is an incredibly inhospitable time. I gave a survey one time where people had to type it in, but basically, what about early early Earth would make you not want to live there. And the most written down answer, it wasn't multiple choice, people wrote it down, was that there was no Wi-Fi. So no oxygen, no Wi-Fi, no way to live here. You would die within seconds. So how do we get from that to a world outside where there is life absolutely covering everything? There is, everywhere you go, from the bottom of the ocean to the top of mountains, you find at least microbial life, little tiny things that you need a microscope to see. You find them everywhere. So we know from looking at asteroids, and we know from looking at places where there is not life now and never has been, that you can have 4 big things in place and you can start to build monomers. And remember, monomers are the building blocks of polymers, and polymers, when they're put together correctly, inside of a phospholipid container will become a cell. You just need a couple steps. So bear with me because there's a lot of time involved in this, but we're going to go through the steps of abiogenesis, life from not life. And I think my head is covering this over here, but this says A is not, bio is life, and genesis is creation. Abiogenesis means the creation of life from not life. Abiotic synthesis means that monomers arose through things that were not alive, making them form together. So I'm going to use amino acids as an example because we have found them on asteroids in outer space where they are not originating from Earth. We know monomers can form given these 4 conditions. So we hypothesize that those conditions must have existed on early Earth. The big one here is we need little or no free oxygen. Remember, oxygen here is incredibly electronegative. This is from a couple lectures ago. That incredibly electronegative oxygen pulls electrons from other things. It's the idea that we talk about antioxidants. Maybe you have to eat blueberries or you rub something on your face to stop aging and the degradation of your cells. What you're fighting is you're trying to fight against oxidation. Oxygen drives oxidation, the breaking down of molecules. So when there's a lot of free oxygen, just this electronegative element, what will happen is bonds will tend to break apart. So in order for me to spontaneously build bonds, to build bonds without life involved, I need to lower the amount of oxygen. And so think about now, and it's always good because I make everybody wait in lecture when I do this and no one can stand it. But right now, before we go on, write down in your notes, if that's true, and early Earth had little or no free oxygen, we have 21% oxygen in our atmosphere today. It's pretty abundant. That's what we're breathing. We're drawing it in and we're doing our cell respiration with it. Where did it come from? Everything is making noise today. I have recorded this a couple times. I'm just going to keep going unless it gets really bad. I hope that it's not interfering in the volume of this. So we have this no oxygen or low oxygen, and I don't mean that the oxygen is bound up in water. There could be water, but it's not oxygen gas that's available out there. And we have this, I move them over for you. We have this atmosphere with low oxygen and a lot of energy. There is a lot of volcanoes going off, meteors striking, lightning striking in the air. The environment is incredibly unpleasant. It's very unstable. It's very energetic. And that energy is going to help drive those molecules to bump into each other. So maybe the water is boiling here and the soil is boiling here, and we're creating a lot of chances that the molecules are going to come close together, that maybe some carbon and some hydrogen are going to be able to form a covalent bond between them. We're starting to have maybe just molecules that are able to interact and approach each other. We got to have those molecules, Sean. You're going to have carbon, hydrogen, oxygen, nitrogen. Some phosphorus would be good. Some sulfur would be good. The building blocks of all of the macromolecules that we talked about in topic number 3 We're going to have all those in place because they're not going to arise magically from nowhere. They have to be there for all this energy to cause them to maybe wiggle and get close and form bonds. And if I want to form bonds, I can't have oxygen interfering by breaking them. So I have a low oxygen, high energy, and my building blocks are all there. We know that water came in from comet strikes and comet tails. There's other sources of water. There are inorganic minerals, atmospheric chemistry, atmospheric chemicals. These things came to Earth either during the solidification of the Earth for more than 4.5 billion years ago, or they came during meteor and asteroid strikes. Comet strikes, too. And finally, time. The Earth is 4.6 billion years old. And when I say that it's that old, that means that as this solar system formed, the solid material formed into planets about 4.6 billion years ago. Earth became a solid blob. That does not mean it became something like today. It was rock with a little bit of energy, and it's very unstable. We don't see life show up until 3.5 billion years ago. And you're like, oh, it's like a 1.1 billion years. It's not much. That's a lot. A billion is a lot. It's a lot more than a million. It's a lot more than 100. In this time, molecules bumped up against each other. They interacted. There was energy. And it caused, and all we're saying is happening is that carbon and hydrogen, maybe some nitrogen, ended up forming a fairly more stable molecule than the elements alone. So we went from just Chon and instability, no oxygen, and time, we end up forming something that we recognize today as the early amino acids, the very basic 4 amino acids. We find them on asteroids in outer space. We also find them in other places where we don't expect to find life making them. They seem to build as a more stable version instead of having those elements just around by themselves. We have 2 separate hypotheses that I'm going to show you. These are not the only 2 hypotheses, but there are 2 hypotheses suggesting how this could have happened. Like, how do you go from just atoms to monomers, something like an amino acid? The oldest version of this, so the first hypothesis that was really kind of solid and was grounded in how we understood the formation of the Earth to work, is called the prebiotic soup hypothesis. It suggests that spontaneously monomers formed on the surface, on land, on the surface of the Earth, because the conditions were right. They were the result of appropriate conditions. And it's prebiotic soup. It's sort of saying that it may have been at the edge of some freshwater lakes, maybe at the edge of the oceans, somewhere where the water and the land came into contact with each other. It means that the atmosphere of Earth must have been very low oxygen. It means that this took a lot of time. Now, I want to introduce 2 people here. This is Operin and JBS Haldane. This was originally their hypothesis in the 1920s. I'm introducing their names here not because you have to memorize them, but they come up a lot, especially JBS Haldane for me, because he made a lot of inroads into some entomological studies. Operin as well. We'll talk about them in other places. So I'm creating some chunking in your brain to start. When that prebiotic soup hypothesis was first hypothesized in the 1920s, everyone was like, that's great, but how are you going to test it? Time machine? Find another planet? And even if you do find another planet, and it happens, you sit there and watch it happen over 1 billion years, there's no reason to say that that's what happened on Earth. That's what happened on that planet. So people were struggling with the idea and had kind of given up on the idea that this could ever be tested. In fact, it was famously called the untestable hypothesis, which we know hypotheses need to be tested. They need to be verified. They need to be reevaluated. You can't just have, it's not science. You're just making guesses out there then. So there was a grad student who was sitting in class and his professor was talking about the prebiotic soup hypotheses, and it was at the time their best hypothesis. And Stanley Miller, who was the grad student, was like, I can do that in my lab. He's like, I can build that. So he built essentially a distillery. This is how alcohol is made. You take some alcohol and a liquid and a lot of water, and you boil off the water and you collect the alcohol. He tested it without alcohol. He tested all the things that Operon and JBS Haldane said had to be there. He put all the kind of ancient ocean things in the water here. He added energy. So he put chon and a lot of energy. He boiled it, and he caused those molecules to shake. They in the water that was in here, they turned into a gas. They flew up into the atmosphere. He applied some lightning, because there must have been a lot of lightning. Methane, water, hydrogen gas, all of the primitive atmosphere gases he had floating around in this gas chamber. And then he cooled it, and he allowed the what was produced to condense into a collector. And he ran this for quite a bit. And when he ran it long enough, he found that there were amino acids and other organic molecules, things that initially we thought only life could make. He found them in the trap. This is important. He did not create life in the lab. We would be having a very different discussion if Miller turned on an experiment and created living organisms. Miller showed that molecules like amino acids can form without living beings creating them. They can form without life. And since then, and if you're really interested in this, if you can take honors evolution, or there's a couple different evolution classes that happen over the course of several years, but sort of discussions in evolutions and theory in evolution. A lot of them talk about these experiments and how they apply to other aspects of the world, things we see in the ocean, things we see in other places where these molecules are spontaneously created. Miller created an experiment where he showed that prebiotic soup hypothesis was possible. It was one piece of support for a hypothesis that had been suggested 30 years prior. Now, there's a problem with this, because when we look at, and we go back more recently, and we've looked at records in the rocks that show that we can analyze how much oxygen was in the atmosphere, we find that there was a lot of oxygen in the atmosphere. So the thing that Operon and JBS Haldane said was required to prevent bonds from being broken apart, free oxygen, was abundant. It was just a little bit less. We think like 17%, but a little bit less to today, but enough that it's could have broken bonds, which has opened up inroads into a lot of other hypotheses. I'm just going to talk about one. I'm going to talk about these because there are other classes later that will pull these apart and compare them. In fact, today, most people don't think the prebiotic soup hypothesis is very valid anymore. We teach it as the very understandable beginning. There's a lot more support for this one. It's called the iron-sulfur world hypothesis. And it hypothesized the same 4 building blocks: low oxygen, time, chon, and an energy source were there in early Earth, but it says it was happening at the ocean floor. Because at the ocean floor, there is still very low oxygen. And we can see these molecules forming in the absence of life. These are hydrothermal vents. That means water and then heat. These are hot vents underneath the ocean. They spew out heat. And they're extremely well studied. In fact, we at Rutgers, we have a series of submarines that goes down and looks at them. The University of Delaware also has a submarine program that goes down and studies these hydrothermal vents. And you've probably seen movies about like the weird life under the sea. These things, when they spew out chon and other basic building blocks, iron particularly, and I don't know if you remember from chemistry, but iron is a really good catalyst. It rusts. It really easily gives up electrons. It's really good at like building things together. Catalysts cause reactions to happen with less energy than you would expect. It makes it easier for them to happen. When I have these hydrothermal vents, I have some iron, I've got some sulfur, I've got some heat. I've got low oxygen, and I have time and lots of chon. Building blocks can arise. Not life. Amino acids. And we do find things, it's crazy. We find 4 different kinds of amino acids on their own with not attached to anything. But we also find some proteins that we only find later in cells. These proteins are folding and existing in the absence of life. They are just molecules. Super cool. I want to change this slowly so we can make sure to see it. Whether we got our molecules from prebiotic soup, probably not. Iron sulfur world, maybe. There's still some other hypotheses that you're going to look at in later classes. But however I got my monomers, the next thing I want to do is build them together. I need to polymerize them or make macromolecules. Now this is easy. We don't even have a special hypothesis for this because we can do this. What is this called? I take one monomer and I make another monomer and I run some kind of reaction that's here, and then I end up with a polymer. What do I remove from these? Water, right? If you go lay out on the beach on a hot day, what does the sun remove from you? Water. This just happens. I can put these amino acids on a rock and so someplace hot, sun shines on them, they'll actually snap together. The water will remove and they'll come back together. This works with amino acids. It works with the building blocks of RNA, which we'll come to in the second part of the class. They just polymerize based on environmental situations. Now, this is not optimal in a living organism. It takes a long time to do this. You would die before you built all your molecules together if you just waited for dehydration to happen. You add enzymes, but this is a process that will happen if I add enough energy to the system. We have some negative ions. They'll help bind these together, and I end up with polymers. Now I've got polymers, and I'll leave you to later classes to discuss protein or RNA, but it was one of these. I lean toward protein. That's my bias, though. You'll have other professors that'll tell you it must have been RNA. Whether it was protein or RNA, a polymer formed. It's not doing life stuff yet. It's just a big molecule. I got to put it somewhere. You are not made out of just proteins and RNA and DNA and molecules all just flapping around. You take all of your important macromolecules and you put them in little containers. Made out of a ring of phospholipids, you build yourself a cell membrane. And every living organism on Earth has cell membranes made out of those phospholipids we discussed in the last lecture. Well, it turns out phospholipids are abundant in the ocean. They spontaneously form. There's a lot of phosphate. It erodes from rocks. Hydrocarbons form just chains of hydrogen and carbon because they bump into each other and they form bonds. And so it's very common to just find these. These are called protocells. They're not cells. They're not alive. They're fat balls. But they're hydrophobic. And they're made of those phospholipids. So what I end up, instead of like a blob of butter, I end up with a shape where it is a hollow globe. When these form together, they make a blob, and they have a space inside that can trap water, and water cannot pass through that phospholipid bilayer. So there's water inside and there's water outside, but there's a container. Even neater, if something got stuck in the protocell, like an amino acids, many of them, the early ones, the simple ones, are all polar. Can they just pass through a fat membrane? No, they dissolve in water. They're hydrophilic, which means they can't get through the membrane. The membrane pushes them away. So if I end up forming one of these with some amino acids inside, now I have a fat ball with some amino acids inside. Now that's as exciting as it gets. Protocells form. They form with some monomers, some polymers inside. Now, you'll look at these in some of your labs. They're actually really fun to make. You pour the phospholipids in, you whip it up with a spoon. They'll actually form these protocells. And what's really strange is they do some of life's processes, things that you think only life does. They go through osmosis, meaning water draws across this membrane if it's got a solute inside. And if that's new to you, I have a whole lecture on it in 2 weeks. So just kind of underline osmosis and say we will get back to it. They have homeostasis. That means they keep themselves about that right shape. They tend to keep that stuff inside of them. And they look like they're maintaining themselves in the face of maybe an environment that is different. And if they get big enough, they pop in half and make two. But the thing is, the two of them are not identical. Maybe this big fat ball here, this big protocell, gets so big that it actually just splits down the middle and it makes two smaller ones. This one has different stuff inside than this one. And that means they are not alive. They have no way of passing on their own characteristics. They do some life stuff, but so does the Roomba. The Roomba moves around. The Roomba, maintains itself a little bit. It even uses up energy. It eats electricity. But it doesn't make baby Roombas, which is good or bad, depending on how you feel about Roombas. Without this mechanism of heredity, without being able to make copies of itself, it's not alive. So we have the monomers. We had two hypotheses that how they formed. Both hypotheses required the same stuff, but had slightly different ways they were generating monomers. The monomers snap together to form polymers. We get a chain of amino acids. And then you put those amino acids in a ball, and time passes. And remember, this is 1.1 billion years that passes. At some point, those protocells become cells when they are able to copy the material inside their cells. They had monomers, it formed polymers inside of a container, and then that at some point they were able to accurately pass on a copy of what their insides were. They gained a mechanism of heredity. And there is an entire course, Early Evolution, at Rutgers that spends about half their time talking about this. Honors Evolution 2 spends a lot of time talking about this. This is from an old textbook. I have tried to put these in here, but if you have an old Campbell textbook, it shows these are the figures from that. Your textbook, the free one we have, the Biology 2E, it's missing this topic, and we really decided that we'd rather still give you this topic. So no requirement to look at this figure, but if you have it, it's this one right here. I'm going to start out by saying that we hypothesized that the first way that cells could copy their information was not DNA. DNA is really complicated, but a very simple strand of RNA. RNA is like DNA, except it is single-stranded. DNA is the mechanism you use to pass your heredity onto your offspring. RNA is good enough if you're a single-celled organism that has very little going on, but it's also easy to make mistakes. It breaks easily. It's difficult to copy accurately. It's not as good as DNA, but it's good enough to be life. The other thing that RNA is capable of is, in a few cases, and we'll see them much later in the class, RNA is capable of acting like an enzyme. It can help cause proteins to follow instructions and be built in the same way over and over again. We call those enzymes ribozymes. They are RNA enzymes. RNA does everything you need to be a basic life living organism. But today, all life has DNA. DNA is like 2 RNAs and 1 mutation, more complex than RNA. During the second part of the class, so after exam 1, I'm going to delve into DNA and RNA and what makes them so special. I'm asking you to put a tag in your notes here. We are going to come back to this slide. We're going to come back to this concept and then kind of head off in the discussion of nucleic acids and how important they were. The same way in lecture 3, I said I'm not going to talk about DNA and RNA yet. We'll come back to that again. So this is a topic for maybe a month from now. I will leave you with this practice question. There is only one more video from this. This is a short lecture. This practice question is asking you about abiotic synthesis and the prebiotic soup hypothesis. I want you to know what stage of abiotic synthesis was facilitated by low oxygen. Don't know if I actually said that. I think I hope I danced around it, so you have to think through it. In order to answer this, I think your best strategy is to first build a sequence of abiogenesis, go through and talk about like build the 4 things you need, and then how I would have had to have put them together, what was needed in each one of those basic requirements to follow through the steps of creation of a monomer. And then I think you can answer this easily. I will come back in the next part of this lecture. There's only 2. And I'll see you soon. All right. Bye-bye.